Hi everyone, good afternoon. I will now present some results of the endTB clinical trial, which is part of the endTB project, which was presented by Mathieu just before. And more precisely, I will present the results in key drug-resistant TB patient subgroups. So a bit of a background. Back in 2015, multi-drug-resistant tuberculosis represented nearly 500,000 of cases and was responsible of 190,000 deaths per year. At that time, the treatment was long, between 18 to 24 months. It was toxic, complex, required taking a big amount of pills every day, including injectable from 6 to 8 months. And it was expensive, and on top of that, it was poorly effective with a success efficacy ranging from 50 to 60%. So the arrival on the market of bedaquiline and delamanid a few years before offered the opportunity to change this treatment to all oral and more user-friendly regimen. So that's when the endTB project, that includes the clinical trial, was implemented with the aim to improve treatment of MDR-TB and find better, shorter, and less toxic regimen. So the endTB trial, so it was a randomized, controlled, open-label, non-inferiority phase 3 trial that included participants of 15 years old with rifampicin-resistant, fluoroquinolone susceptible pulmonary TB. The objective was to compare each of the 5 experimental regimens to the control, and the treatment duration in the experimental regimen was 9 months, while it was approximately 18 months in the control arm. Patients were followed for at least 73 weeks, which is 17 months, and up to 104 weeks, which is 24 months. And the primary endpoint was a favorable outcome at week 73. So all 5 endTB experimental regimens that are presented here, they all had at least one new drug, being bedaquiline or delamanid, one repurposed drug being clofazimine or linezolid, all had fluoroquinolone, which is moxifloxacin or levofloxacin, and all received pyrazinamide. The control arm was a current WHO standard of care, so it may contain all the drugs that were currently recommended by WHO, and in particular, it may contain the new and repurposed drugs that were composing the experimental regimen. Now the endTB clinical trial is terminated and the results have been presented recently, so we will quickly go through those main results, but the objective of the subgroup analysis that I'm presenting today was to explore the efficacy result of the endTB trial in key subgroups. Do these results hold? Can we find any signals in some specific populations? And also, because several short regimens are now available, can we help clinicians to make the choice between the different endTB regimens for the patient with a more difficult to treat form of disease? So for the population, we were interested in baseline characteristics that could influence the choice or the success of any experimental regimen. And the risk factors were selected from the literature, keeping the ones that were shown in previous study to potentially affect or change the effect of shorter regimen compared to longer ones. So in this way, we kept the following baseline characteristics: HIV co-infection, low BMI, diabetes, and severe disease. In terms of method, we look at the proportion of favorable outcome at week 73 in all the participants who had this characteristic, and also the participants who did not have these characteristics in all experimental arm and in the control arm. The risk difference was calculated as a difference of proportion of favorable outcome between each experimental arm and the control arm, along with the 95% confidence interval, using a Poisson regression model with robust standard errors. The population that we used for this analysis was the modified intent to treat, or MITT population, which is a population that contained all the randomized participants who received at least one dose of study treatment, who had the culture positive, who were shown to be resistant to rifampicin, but were not known to be resistant to any experimental arm drugs, excluding pyrazinamide. We will focus in this analysis on the arms that were found to be non-inferior in the main study. And we precise here that this study has to be seen as an exploratory analysis, as the study was not powered and not designed for this comparison. Okay, so the patients were recruited in 12 sites in the endTB clinical trial, over 7 countries all over the world, as you can see here on the map. The 754 patients were randomized as initially planned between February 2017 until October 2021. And the database was locked and the analysis started in June 2023, so last year. Here you can see the proportion of favorable outcome at week 73 in all arms. So the main thing that we see, it's the high proportion of favorable outcome in all arms, ranging from 78.8% to 90.4%. And an important thing to note here also is the high proportion also favorable outcome in the control arm, which is performing very well with over 80% of successful outcome. So 3 arms in the endTB clinical trial, so endTB 1, 2, and 3, were shown to be non-inferior in all analyses, and endTB 2 was even superior to the control. Oh, j'appuie sur le truc. Right. So now we will look at the results in the subgroup of interest, and all subgroups will be presented in the same way, so on a forest plot like this. So for each arms that were shown to be non-inferior in the main study, endTB 1, 2, and 3, we present on the first line the result of the overall population. And then for each subgroup of interest, for each category, we'll present all the categories of the subgroup, with the one category of interest being highlighted in blue, like here you can see the HIV positive, for example. Then we have the number and the proportion of favorable outcome in the experimental arm first, and then in the control. And finally, on the right, you can see the risk difference represented as a square, along with its 95% confidence interval as a bar. The vertical line represents a risk difference of 0, so everything which is on the right of this line favors the experimental arm, while everything on the left favors the control arm. So for HIV positive, we see that in arm 1 and 2, result in HIV positive seems consistent with the overall result, while in arm 3, the effect seems to be different in HIV positive compared to the overall population. It favors the control arm. It's on the left of the vertical line. And it also appears to be different from the HIV group that were receiving endTB 1 and endTB 2. Then with our definition of disease severity, we see no difference in the participant with a more severe disease from the overall population in the 3 arms. The general findings of endTB still remain valid in this population, and we cannot see any difference between the three groups for participants with a severe disease. In the case of low BMI, we see that the 3 arms seems to favor the control arm rather than the experimental arm, which was not the case in the overall population, and we don't see any difference between the 3 arms. If we finally look at the patient with diabetes, the results seem to suggest also differences in treatment effect from the overall population, but in favor this time of the experimental arm. So even if the confidence intervals are wide, the direction remains the same from the overall population. And we don't see any difference between the 3 arms. So as a conclusion, we did not detect any signal in patients with severe disease or diabetes, so it seems reasonable to use endTB 1, 2, or 3 in these populations. While we detected a first signal in HIV-infected patients in arm 3, so it might be preferable to use endTB 1 or 2 over endTB 3 in HIV positive. And then we detected also a second signal in patients with low BMI in all 3 arms. So for these patients, best treatments are still unclear, and maybe longer or regimen with more drugs could achieve better results. So we think that this first element can be already included in the discussion with patients to inform and guide the choice of treatment. And of course, further evidence and additional research are needed to confirm or not these signals, and also to increase knowledge to get clinicians on the choice of regimen for this subgroup of patients. We would like to thank all the people who have been involved in the endTB clinical project. So as you see, it's a lot of people, a lot of partners. And we also would like to thank all the participants, the team on site, the central team, which are a lot of people that are here now. And thank you, everyone, for your attention.